Hi, my name is Stormie Collins, and I'm a PhD student in Dr. Heather Bracken-Grissom's lab at Florida International University. Today I'll be presenting on the complete mitochondrial genome of the pink shrimp Farfantepenaeus duorarum. This study was conducted with Dr. Heather Bracken-Grissom and Dr. Antonio Baeza from Clemson University. Farfantepenaeus duorarum is a commercially harvested penaeid shrimp found in shallow coastal waters. This shrimp is very abundant and is economically relevant as it is heavily targeted for use in bait and seafood consumption. In 2020, over 600 tons of Farfantepenaeus duorarum were collected in the Gulf of Mexico, totaling over $5 million in commercial revenue. The objectives of the study were to describe the complete mitochondrial genome, to assess gene order, nucleotide composition, and codon usage profiles, to analyze the selective pressure constraints, and to describe the secondary structure of all tRNA genes and within the D loop. The raw sequence data used for the study was obtained from previously generated data from Wolf et al. 2019. The dataset was composed of over 5 million paired reads, and the mitochondrial genome was assembled de novo using the Novoplasty software. The mitochondrial genome was annotated using the MITOS and MITOS2 web servers. Using MegaX and the XPaSI web translator tool, each gene was then manually curated. Genome VX was used to render a visual depiction of the mitochondrial genome. MegaX was used to calculate nucleotide composition and codon usage, and Sequence Manipulation Suite's codon usage tool was used to determine codon usage of all protein coding genes. The control region was examined for microsatellites using BioPHP Microsatellite Repeats Finder and for tandem repeats using Tandem Repeats Finder. The Vienna RNA Web Services tool was used to visualize the secondary structure of each tRNA. The RNA Structure Web Server was used to analyze the control region for secondary hairpin structures. Finally, we performed a selective pressure analysis for each protein coding gene using Ka/Ks calculator based on a comparison between F. duorarum and F. californiensis. The mitochondrial genome of Farfantepenaeus duorarum is 15,971 base pairs in length and is comprised of 13 protein-coding genes, 2 ribosomal RNA genes, 22 transfer RNA genes, and a 982 base pair region that is assumed to be the D loop. There are 22 tRNA genes ranging from 65 to 70 base pairs in length, of which 21 were found to exhibit the classical cloverleaf shape. Serine 1 was the only tRNA to lack an arm, and the asparagine and phenylalanine tRNAs were found to display an extra loop in the secondary structure. The selective pressure analysis resulted in Ka/Ks values less than 1, indicating that all mitochondrial protein-coding genes are evolving under purifying or negative selection. The COX-1 gene was found to be under the greatest amount of selective pressure. The mitochondrial gene order is identical to congener F. californiensis. However, tRNAs L1 and L2 were originally reported in an opposite order. Reannotation of the F. californiensis genome found that the loci locations for L1 and L2 were congruent among the two species. Nucleotide compositions equal roughly 35% thymine, 19% cytosine, 32% adenine, and 14% guanine. The overall AT content was higher than CG content, as is consistent in other penaeid shrimps and with our codon usage analysis, as the least used codons were CG heavy. 7 of the 13 protein coding genes begin with the traditional ATG start codons, 10 protein coding genes end with the traditional TAA stop codons, and 2 genes were found to end with an incomplete stop codon. All protein coding genes were found to be under negative selective pressure. COX-1 was found to be under the greatest amount of selective pressure. Mitochondrial genes have long been thought to be under neutral pressure, and our findings suggest that CO1 is evolving under purifying selection. We suggest that future studies examine the effect of selective pressures when using CO1 and other mitochondrial protein-coding genes to resolve phylogenetic relationships. Within the D loop, 15 AT-rich microsatellite repeats were found, 0 tandem repeats were found, and 20 possible stem loop structures were found in variable lengths and location. This is the first time the control region has been examined for secondary structures for any species within the Penaeidae. In conclusion, the mitochondrial genome of Farfantepenaeus duorarum is 15,971 base pairs in length and is comprised of 13 protein-coding genes, 2 ribosomal RNA genes, 22 transfer RNA genes, and a D loop 982 base pairs in length. Gene order is identical to F. californiensis, and all protein-coding genes are evolving under negative selective pressure. This study adds one more mitochondrial genome to the Penaeidae, which may be useful in resolving relevant taxonomic issues for this economically important commercially harvested species. Thanks so much for listening, and I'm open to any questions that you may have. Bye!